So I'd like to welcome everyone and thank you for joining us today. My name is Corinth Auld and I am Mercodia's Medical Science Liaison. At the end of the presentation, I will read any submitted questions and then our speaker will have a chance to answer them. So with that being said, I'd like to introduce our distinguished speaker, Dr. Alan Cherrington. Dr. Cherrington is currently a professor in the Department of Molecular Physiology and Biophysics, as well as the Department of Medicine at Vanderbilt University, where he has been part of the faculty since 1975. He also holds the Jacqueline A. Turner and Dr. Dorothy J. Turner Chair in Diabetes Research. He has served on editorial boards of highly regarded scientific journals, holds multiple appointments, and has held elected positions such as the 2004-2005 National President of the American Diabetes Association. Dr. Cherrington's work over the years has defined the effects of various hormonal and neuronal factors on glucose metabolism in the liver. For example, he has characterized the effects of insulin, glucagon, cortisol, epinephrine, and norepinephrine on the rate of hepatic glycogenolysis and gluconeogenesis in vivo. He's recognized worldwide as an authority in this area, and we are very happy that he will be speaking to us today about glucagon as a therapeutic target. Dr. Cherrington.
Thank you, Corinne. It's a pleasure to be talking to everybody. Good afternoon or good morning, depending on where you are. The purpose, in my mind, of this talk today is really to discuss the physiology of glucagon more so than its biochemistry, and to really see how that fits in normal physiology and in pathophysiology, namely the diabetic state. So, to begin with, there are really four topics that I'm going to discuss. The first is the sensitivity of hepatic glucose production to glucagon in vivo, and really what I want to point out there is that subtle changes in glucagon can have big physiologic effects. In fact, probably our ability to assay glucagon, it limits our ability to understand its physiology. The second point is that the impact of glucagon on hepatic glucose uptake is also a factor, both in the normal and the pathologic state. So glucagon is not simply a hormone that controls glucose production, which it's been known to do for many years, but it actually also has profound effects on glucose storage by the liver. And then thirdly, we'll look at the role of glucagon in the diabetic phenotype, so both type 1 and type 2. What is the evidence that it's really a key player? And then we'll finish by looking at some fairly new work relating to glucagon as a target for diabetes treatment. Part of what I'll talk about today comes from the work of other labs, and part also comes from our lab, and so I thought it would be worthwhile in that regard to go over some of the methods that we use so you'll better be able to appreciate the data. So we have used the conscious dog as a model for many years, and there are certain advantages to that, not the least of which, if you're interested in liver metabolism, is the ability to put catheters both upstream and downstream of the liver. So as you can see here, the dog is prepared, and this usually happens about 3 weeks before an experiment, and we insert catheters in the femoral artery, in the hepatic vein, and the portal vein, and we also put catheters into a jejunal and splenic vein. And the animal is then allowed to recover, and we study it in the conscious state, usually 2 to 3 weeks after the surgery. Now, because we can put catheters in the blood flowing into the liver and in the blood leaving the liver, we can actually use the AV difference technique to really quantify what the liver is doing. So, as shown on the bottom of the slide, the load of glucose going into the liver, and I'm using glucose, it could be any parameter, but we're going to focus on glucose. The arterial glucose level times the arterial blood flow plus the portal glucose level times portal blood flow gives you the load of glucose entering the liver. The hepatic glucose level times hepatic blood flow gives you the load of glucose leaving the liver, and of course the difference between the two will tell you whether the liver is storing or producing glucose. Now, if you combine the AV difference approach with a tracer approach, you can actually also measure the balance of tritiated glucose or a stable isotope of glucose across the liver. And that will give you the absolute flux in and the absolute flux out. So in other words, We can measure the amount of glucose that is actually going into the liver in an absolute sense, the amount that is coming out in an absolute sense, and the balance between the two gives the net balance. Now, again, the advantage to the dog is that one can get catheters in these places, and that would be true of other large animal models, such as the pig or the non-human primate. Very, very difficult to do in man, obviously, and impossible to do in rodents. So it does give us a unique window on liver physiology. The other advantage shown in the next slide is that when we do pancreatic clamps, we can actually give back the pancreatic hormones into their normal site of entry into the circulation. So because we have those catheters in the jejunal and splenic veins, we can actually infuse insulin and glucagon directly into the portal vein. And so by doing that, when we create a clamp, we actually clamp both the liver and the rest of the body at appropriate levels. So shown here, you can see the normal situation. The beta cells produce insulin, the alpha cells produce glucagon, the liver sees a much higher level of both than do the tissues and the rest of the body. When we give somatostatin to inhibit the pancreas, the insulin secretion is inhibited, glucagon secretion is inhibited, and we replace those secretions by infusions directly into the site that they were being secreted. So if we do this correctly, we can basically take over control of the endocrine pancreas without disturbing metabolism. And this is different from, again, what can be done in a rodent or in the human, where you have to infuse the hormone, or you give the somatostatin peripherally, and you also have to infuse the insulin or the glucagon peripherally. You can't get into the portal vein. And that, of course, disrupts the normal gradient between portal blood and blood everywhere else, and you have to take that into account in interpreting the data. So it's another little advantage that one has when working in a large animal model. So armed with these techniques, we undertook studies to, first of all, assess how potent my trigger on my computer is. Kind of tardy, so you may have some slow appearance of slides. But first what we did was want to investigate how sensitive the liver was to a rise in glucagon in terms of its ability to produce glucose. So these data come from a study in which there were two groups. Shown on the left in yellow are control experiments. Shown on the right in orange are the test group. And what you can see immediately is that the insulin level in the artery, the circles, and if you look at the data on the left first, that the level in the portal vein is about 3 times the level in the artery. With glucagon, it's not the same ratio, but clearly the portal levels of glucagon are much higher than the arterial. I would also point out that over time, glucagon assays have changed. I don't think any of them have ever precisely measured glucagon. The assay that we have generally used also has changed over time. Back when these experiments were done, about 25 picograms per mil of the level there was actually not glucagon. It was some cross-reacting junk. So in essence, the ratio of portal glucagon to arterial is about 2 to 1. As opposed to insulin, which is about 3 to 1. And I keep that in mind because that will be important later on. If you look over to the right, you can see that in this set of experiments, we used the pancreatic clamp to keep glucagon basal, so the arterial and the portal insulin levels are basal. And you can see at the bottom, the glucagon level was increased 4-fold. So both the arterial and the portal vein glucagon levels were elevated about 4-fold. So the question is, this physiologic change in glucagon will do what to glucose production? And there's the answer. So if you look at the left, the lower left, you can see the AV difference-determined hepatic glucose output. So this is net hepatic glucose output by the liver. And if you look at the glucagon, the orange, you can see that there is a very quick rise. In fact, the time to half activation with glucagon is about 8 minutes. One of the characteristics of the hormone is its quick ability to respond to a challenge and provide glucose for the rest of the body. And you can see that clearly manifest here. Equally obvious is the fact that it does not have a sustained effect. So the increase in net hepatic glucose output drops over time. And in fact, by 3 hours has been reduced by probably 70%. Nevertheless, the increase in glucose production causes hyperglycemia to occur, as you can see in the top panel. And in this case, it reaches about 160 milligrams per deciliter. Now, if you infuse glucose to parallel that, the yellow data, you can see that hyperglycemia itself inhibits net hepatic glucose output. So if you look again at the bottom, you will see that the yellow line tends to trend towards the axis. So the actual effect of the glucagon rise is the difference between the orange and the yellow line. And clearly the effect is rapid, but it is evanescent. But nevertheless, even at 3 hours, there is still a substantial effect of the rising glucagon. So it's not that if its effect disappears, the liver just recalibrates. So the next question is, what is the source of the glucose that the liver is producing? Is this a glycogenolytic or a gluconeogenic effect? And without going into detail of the methodology, I think the data on the right point out pretty clearly that it's a glycogenolytic and not a gluconeogenic effect. If you look at the top panel, you can see that the rise in glycogenolysis occurs within minutes and again drops over time, but is still evident even at 3 hours. On the other hand, there is very little, if any, change in gluconeogenesis. Now, this might seem strange considering glucagon is known to affect gluconeogenic gene transcription in the liver. But you have to remember two things. First of all, changing liver gluconeogenesis will only be effective if you can supply substrate for that process. And since glucagon can't do that, basically you can rev up the engine, but once you deplete the gluconeogenic precursors in the blood, you're going to go right back to the same gluconeogenic rate that you had in the beginning. The other thing is the gluconeogenic effects on gene transcription, while they occur rapidly, don't result in significant changes in protein for 2 to 3 hours. So you also wouldn't expect to see much of a change in gluconeogenic flux here. So bottom line, glucagon is a very sensitive stimulator of the liver. It works very quickly, but its effect declines over time, and that effect is primarily glycogenolytic. So the next question is, how does this apply to the human? And again, the data here look at studies that were carried out in the human just over 10 years ago. And I'd like to point out at the top, they used the pancreatic clamp technique, but it has the limits that I talked about earlier. So when you look in the control period, so the minus 30 to 0 time, you can see that the arterial level and the portal level are about the same. So they gave somatostatin, which inhibited insulin secretion, and they gave back insulin to clamp the arterial level. Because insulin secretion was off and they could not replace intraportally, the intraportal insulin level, instead of being 2 to 3 times what it would otherwise have been, it's now down near the arterial level. The other thing is that when you use somatostatin in the human, you can't use as much as is used in the dog, and so you can't get a complete inhibition of insulin secretion. And so what you see happens during the glucagon infusion when hyperglycemia occurs is you actually get a bit of breakthrough and a bit of insulin secretion. So ironically, you end up with a relatively normal arterial to portal insulin ratio. It's not quite as high as you would like it to be, but nevertheless, it's a pretty reasonable approximation. And so if you now look at the top on the right, you can see that the glucagon in this case went up to about 225 picograms per mil. So here you have a rise in glucagon in the human on the background of what is a reasonably basal insulin environment. And you can see if you look at the right again in the lower panel, there's again a very rapid increase in glucose production, which falls over time, and that creates a marked hyperglycemia shown in the lower left. the only point I want to make here is that the human also responds very fast to glucagon. It is also evanescent. Now, they didn't break it up into its glycogenolytic or gluconeogenic components, but my guess would be that it would have been, in fact, glycogenolytic. Now, one of the things that comes up from time to time is that most people, because of the ease of doing the experiment, use square wave infusions of insulin, glucagon, or whatever. And does that really make any difference? And so we carried out a study a number of years ago to actually address that question, and the results are shown here. So there were two groups. In one group, the glucagon infusion rate was quadrupled in a square wave fashion. If you look at the upper left, that's the orange line that goes through the white oscillating line. And it was also given in a pulsatile fashion to correspond to the normally occurring synchronicity of insulin secretion. So the period and the range of the oscillation really reflect a general observation that this is what glucagon on a secretion on a minute-to-minute basis looks like. And so the question is, does it matter if glucagon is oscillating or square wave in response to this rise? And again, this was done on the background of a pancreatic clamp where we kept insulin flat, as you can see in the lower left. And what happened, of course, is, as shown in the right side of the panel, is both glucose production and the ensuing rise in glucose were absolutely identical, regardless of whether the oscillation was there or not. Now, if you take a really close look at the data, you can see some entraining of glucose production by the oscillating waveforms. In other words, there is a little wobble to it. But if you consider area under the curve, the response over, in this case, 4 hours, it made no difference how we infused the hormone, whether it was square wave or whether it was in an oscillating waveform. So we think that the pattern of alpha cell secretion is more a function of the engineering of the alpha cell and does not have any particular biologic gain. It's possible that when in synchrony with oscillating insulin, it becomes abnormal, that might exacerbate some of the problems associated with abnormal insulin secretion. But that's not very clear at this point. Okay. So what happens when you do the reverse experiment, and that's to say take glucagon away? Or put another way, how important is it when you get up in the morning that glucagon is there driving glucose production? And so what you see on the left, again, are control data. Nothing new. They're similar to what I showed you earlier. But the data on the right indicate what happens when you change glucagon. So the top panel, again, shows that we've clamped insulin both in the artery and the portal vein. But on the bottom, what we do is we turn off the glucagon infusion. And you can see there's a very rapid decline in both the arterial and the portal glucagon levels. So first of all, this is indicative of the speed with which glucagon is cleared from the body. The other thing, the baseline level reached is just under 20 picograms per mil. This is the cross-reacting junk that I talked about. So if you take plasma from this animal, put it over a sizing column, there is no 3,500 molecular weight glucagon there. So we've created a sort of instantaneous, complete glucagon deficiency. Now the next slide shows then what happens. And if you look again at the left lower panel, this is net hepatic glucose output once again. If you look first at the orange line, you can see that glucose production falls from about 2.4 down to basically 0 over the course of about 3 hours. We infused glucose to maintain euglycemia. In the control animals where we did nothing, you can see euglycemia was spontaneously sustained, and you can see very little, if any, change in net hepatic glucose output. So lacking glucagon, then, is the difference between the yellow and the orange line. And what this tells you is that basal glucagon is what is maintaining resting glucose production and supplying the rest of the body with glucose. So it puts it in a very powerful control position. Again, if you ask the question, is this a glycogenolytic or gluconeogenic change, you can see that the answer is it's primarily glycogenolytic. So the data on the right at the top show a rapid and almost complete inhibition of glycogen breakdown. The data at the bottom show a very small decline in gluconeogenic flux. So 80 to 90% of the response is glycogenolytic. So not only does glucagon work sensitively when its level rises, it also works sensitively when its level drops, so that it has a profound effect on glucose production over the whole range of its biologic levels, from 0 all the way up to 200 or 300 picograms per mil. Now again, is this a human phenomenon as well? And I show you data from John Lillienquist's lab many years ago, actually. And here they had the normal human beings, and I would point out, if you look at the top panel on the left, this is the arterial plasma insulin level, and those are the kind of darker yellow, and you can see that they had basal levels. Then they did a pancreatic clamp, and they tried to match those relatively effectively. What I've done here is put on what would have been the portal insulin levels, so that the pale yellow are not measured levels, they're estimated levels, so that prior to the clamp, the insulin level would have been up threefold in the portal vein. And of course, once they clamped, that would have come back down to the arterial level. So in terms of the liver, there's a mild deficiency of insulin in this protocol, unavoidable because of the need to replace insulin peripherally. And what they did was not replace the glucagon. And you can see this was an assay done in 1977, probably with the old Unger 30K antibody, and the insulin, the glucagon levels, I should say, were very high. So don't get confused by the absolute levels. This is a different assay system. But what you can see pretty clearly is there was a dramatic drop when the somatostatin inhibited the glucagon secretion. So what we have is glucagon deficiency in the presence of a mild insulin deficiency. So the insulin deficiency should cause glucose production by the splanchnic bed to increase, whereas glucagon deficiency would drop it. And you can see by looking at the lower right panel that splanchnic glucose production dropped quite dramatically, meaning the drop in insulin was clearly more relevant than the slight defect in insulin secretion. But again, this proves that, in fact, basal glucagon is equally important to sustaining glucose production in the human. Splanchnic glucose production, by the way, just means that they have an arterial blood sample and a hepatic vein, which you can get by passing a catheter through the heart and wedging the catheter into the liver. But you can't get portal. So it represents a synthesis, really, of what is going on in the gastrointestinal tissues and the liver, but it's primarily reflecting the liver. And of course, they clamped the glucose level. So these data extend the data that I showed you to the human and point out that it's really quite important in the human as well. Now, if you bring everything together and you try and create a dose-response curve between glucose production, and in this case it's tracer-determined, this is absolute flux out of the liver, versus the sinusoidal glucagon level, the level that exists when the portal blood and the arterial blood mix, what you see is the response shown here. The data in orange are data from the canine, from our lab. The data in yellow are data from several labs conducting human studies. And what is clear is that there is a really dramatic dose response, so that the power, the control strength of glucagon is very high. You can get up to glucose production rates as high as 10 mg per kg per minute. The blue box is meant to represent the physiologic range as opposed to the pathologic or pharmacologic range. And you can see that it has a level of about 200 as its extreme. To relate that to an arterial level, you divide by 2. So what this is really showing is the effect of 0 glucagon up to 100 picograms per mil in arterial blood. And what that's doing, there's almost a linear relationship over that range between glucagon and glucose production. And it turns out that with assays that are now commonly used. In effect, a glucagon level of about 10 picograms per mil will cause a glucose production rate of about 0.5 mg per kg per minute. Now, as assays become more sophisticated and refined, that number might drop a bit. That is to say, it may take even less glucagon than we think to do that. But the bottom line I want to leave you with is that it doesn't take much glucagon to drive glucose production. And when you think that in diabetic individuals, type 2s, the glucose production rate is anywhere from a half to a mg per kg per minute in excess, that could be caused by 10 or 20 picogram per mil excess of glucagon. So when you look at glucagon levels, don't underestimate the power that they have to drive metabolism, and in this case, glucose production by the liver. So the next topic is going to really focus on uptake, but in order to put in perspective what I just told you in a more broad sense, all I've done here is plot the glucagon levels that are commonly found in 4 circumstances relative to control data from the same studies. And basically what you can see is that there's maybe a 50 or 60 picogram per mil rise in glucagon during exercise. Exercise, you've got to supply glucose to working muscle, and you've got to do it quickly. Glucagon has all the characteristics to do that. And in loss of function experiments, it's been proven that these changes in glucagon are critical to the normal metabolic response. If you look at the lower left, you see what happens during hypoglycemia. We'll talk more about that in a minute. Again, an urgent and quick need for glucose production. Again, a glucagon level of only 30 picograms per mil. And again, that's going to vary depending on the degree of hypo. The top right, infection. So trauma and infection are associated with elevated glucagon in order to drive the catabolic metabolism. Again, a little bigger rise here. This is tending towards the pathologic state. And then fasting, shown on the bottom, again, a difference in the order of 50 or 60 picograms per mil. Now, again, I wouldn't get too hung up on the absolute numbers. They're pretty small to bring about pretty big changes. But glucagon has been shown to be critical to normal metabolic regulation in each of these circumstances. So now, those were circumstances where you need to drive glucose production. What about where you need to drive glucose uptake by the liver, so a meal feeding situation? Does glucagon have any role in that circumstance? And shown here are data from a set of experiments that we carried out a number of years ago to address that question. So if you look at the design, there was a 2-hour equilibration period and a 90-minute period in which we gave glucagon and insulin intraportally through those catheters to keep their levels basal. But we doubled the glucose level by infusing glucose through our leg vein. So in that 90 minutes, we have isolated hyperglycemia. And we know from other studies that that should put the liver into neutral. It should just put it to a 0 production. Then what we do is we create a meal feeding situation. So we raise insulin 4-fold. We give some glucose intraportally to make the brain think that there's a meal being consumed. We continue to infuse glucose peripherally so that the glucose level reaching the liver is doubled. And then what we're going to do is 2 different things with the glucagon. We're either going to double the basal glucagon rate, the orange group, or we're going to have it and then progressively reduce it a little further. And the question is, under this feeding circumstance, is the amount of glucagon that accompanies those feeding signals to the liver going to make any difference to its ability to take up and store glucose? And the next slide shows the arterial blood glucose level on the top in the 2 groups. And as you can see, in that 90-minute pre-period, we were able to double the sugar level, and that was sustained throughout, no difference in the 2 groups. The insulin level initially was basal, then it was increased about 4-fold, and this is arterial. It would be higher in the portal vein. No difference between groups. And there's the glucagon level. This is arterial. Again, basal in that control period. But then when we get to the feeding period, in one group, glucagon goes up, and in the other group, it drifts down. But I want you to take away from this that the difference between those two is probably in the order of 25 or 30 picogram per mil. So the question is, what is that going to do, if anything, to hepatic glucose uptake? And if you look first at the bottom panel, you can see that in the control period, these dogs were producing glucose at a rate of about 2 mg per kg per minute. When we created the isolated hyperglycemia, they switched off. They went to close to 0. Then when we induced the feeding signal, elevated insulin, tricked the brain into thinking it was eating. You can see that you switched to glucose uptake, but you can also see that that glucose uptake is twice as great in the lower glucagon group than in the elevated glucagon group, proving that glucagon not only is a hormone which regulates glucose production, it's also a hormone which can affect metahepatic glucose uptake. Now, again, the question is, does this apply to the dog or does this apply to the human? And there was a study published a number of years ago now from Michael Roden's lab where he actually looked at the impact of glucagon on the liver's ability to take up and store glucose. So they used NMR technology. But let me take you through the hormone and glucose data first. So there's two groups. They're going to create hyperglycemia in both, and you can see it was no different. The plus glucagon is going to be the orange, and the low glucagon is going to be the yellow. They infused insulin, and this again is a peripheral infusion, but nevertheless the insulin level is getting up to 30. Insulin in man shuts down its own secretion to some extent, and so it's a little bit unclear what the portal insulin level would be here. Probably it was slightly elevated, but not dramatically up. But nevertheless, you have a mild hyperinsulinemia at the liver and a distinct hyperglycemia, and what's different here is the glucagon. So in one case, they tried to keep it flat, and in the other case, they allowed it to drop. Again, remember, probably 25 or so in their assay was not real glucagon. So this is a pretty dramatic deficiency. And then what you can see here is the change in liver glycogen measured using NMR over time. And what's clear is in the presence of glucagon, basal glucagon, the rate was much slower than in its absence. So when glucagon was deficient, the liver presumably took up more glucose and stored more glycogen, meaning that the observation that the liver responds to glucagon in an uptake mode is not simply a canine phenomenon, but also applies to the human. So hopefully, so far, I have been able to point out that the liver responds very sensitively, very quickly to glucagon. It's a controller of production. It's also a controller of glucose uptake. Glucagon can affect various tissues in the body. There are receptors in the heart and the gut, but they probably don't function under physiologic levels of glucagon. They respond to pharmacologic levels. There are glucagon receptors in the brain and in fat. I think it's more debatable as to whether those are having a significant role in metabolic regulation. Most of the data suggests they're not. There's the odd study that suggests that maybe they can. I think the answer is not clear there. But of course, on the liver, the answer is very clear. And I want to leave you in this regard with the thought that while I've talked about glycogenolysis and gluconeogenesis, where glucagon and insulin have opposing effects, I don't want you to forget that glucagon can also affect fat and protein metabolism in the liver, and again, in an opposite way to insulin. So it will inhibit fatty acid synthesis. It will promote fat oxidation and ketogenesis. With regard to protein, it will inhibit protein synthesis and enhance protein degradation. So when you think about glucagon and its physiologic effects, I think you should primarily focus on the liver, but within the liver, it can be glucose, fat, and protein metabolism. Okay. So let's now switch to the diabetic state, both type 1 and type 2, and consider what's wrong with the alpha cell. The alpha cell is clearly defective in both diseases. And I show you some data here from Phil Cryer's lab done a number of years ago, where they created a step down in glucose, and that's the top panel. And they did this in non-diabetic individuals and diabetic type 1s. And you can see they step down progressively over time. Shown in the bottom is the glucagon response. And if you look first at yellow, which are the normal individuals, you can see that as glucose was stepped down, nothing much happened until you went below about 70 milligrams per deciliter, in which case the alpha cell began to turn on, and that got progressively greater as the glucose got lower. And so you can see that, indeed, as I alluded to earlier, glucagon is a major component of the counter-regulatory response to hypoglycemia. But clearly, in the type 1 diabetic, that response is defective. It can become defective as quickly as 6 months after diagnosis, and within 2 to 3 years is completely gone in 90% of people with diabetes. So the alpha cell becomes deranged. And I think that it is not completely clear why that happens at this point. It's specific to glucose. If you give arginine to stimulate the alpha cell, it seems to work fine. I think a very interesting area of investigation is to try and understand that, why there's this defect, and I think it would be very helpful if we could come up with treatment strategies that allow it to be overcome, because clearly hypo is probably the most feared symptom and complication of diabetes, and it also limits our ability to lower the blood sugar, frankly. Now, you might wonder how important is that small change in glucagon, and so I want to show you these data. These are more recent data from our lab, where we actually asked that question. And so these, again, are canine experiments, and you can see that we infused insulin in both limbs of the study. In one limb, orange, we're going to allow a normal glucagon response to occur, and in the other, the yellow, we're going to prevent it. So insulin was infused into both groups to elevate the insulin level to induce the hypo. The glucagon, shown in the upper right, was either kept basal or was allowed to respond normally. And you can see this isn't a huge response. It's 25 picograms per mil at the max, 60 minutes, and then it kind of dies a little bit over time. And the question is, what difference does this make to the ability of the body to defend itself? Bearing in mind that it's mainly a liver target, if you look at the bottom, you can see in orange first that when the insulin is there and glucagon is allowed to respond, there is a brief fall in glucose production, which is very quickly countered, and you actually begin to overproduce glucose to offset the actions of insulin. If you don't have the glucagon response, you get the yellow line. You get a much deeper fall in glucose production and a much slower countering. And this countering is entirely dependent on a major release of epinephrine. So the epinephrine levels are dramatically different in these groups. And yet, despite going to the backup system and getting a robust response of epinephrine, the price you pay for not having that small amount of glucagon is about a 10 milligram per deciliter difference in the hypo level. So if you look at the lower left, you can see the yellow line is significantly lower than the orange. So a small defect in glucagon secretion in a hypo circumstance can make a big difference. The other problem is that glucagon is elevated in the basal state, so not only does it not respond properly to hypo, there's too much of it in the presence of hyperglycemia, and that contributes to the abnormal glucose production rate, which contributes to the hyperglycemia. Shown here are data from Roger Unger's lab a number of years ago, where they took type 1 patients and looked at them for 2 days under control circumstance, then gave them somatostatin to inhibit glucagon secretion and then restored glucagon for 2 days. And what you can see on the top is glucose, in the middle is glucagon, and then glucose excretion at the bottom. You can see that they're elevated, the glucose is elevated, the glucagon is very high, and they're excreting glucose in the urine. When you inhibit glucagon, the glucose improves dramatically, there's no glucoseuria. In the control arm, when you restore glucagon close to where it was, you reestablish the hyperglycemia and some glucoseuria. So again, an early proof of concept that in fact, if you could control glucagon in a type 1, you could potentially improve glycemic control. So what about glucose intolerant individuals and type 2s as opposed to type 1? And here I show you some data from IGT patients. And what you can see is 2 groups, so normal glucose tolerance or impaired. They're given an oral glucose load, and then you just watch what happens. So if you look at the yellow first, which is the normals, there's a rapid and robust response of insulin. That's associated with a nice fall in glucagon. Again, look at the numbers, 15 picograms per mil, which is time-dependent. As a result of that, glucose production is inhibited and then slowly rebounds, and you have a limited glycemic excursion. What happens in individuals that are glucose intolerant is interesting. They actually have a somewhat delayed insulin response, but overall they actually secrete more insulin than did the normal glucose tolerant individuals. But the glucagon response is defective. The fall is slow and much less pronounced. Consequently, you see an impaired inhibition of endogenous glucose production that's associated with an excess of hyperglycemia. So again, evidence that glucagon may in fact be contributing significantly even to this phenotype. If you look at a more mature type 2 diabetic model, again, these are healthy subjects or type 2s in red, and they're going to be fed a meal. First of all, you can see from the blood glucose level, these are poorly controlled. The insulins are low. The glucagon is in excess. Again, glucagon is relatively or even absolutely too high in these individuals. And what you can see is in the normals, the gray, there's a minimal excursion of the blood glucose associated with a large rise in insulin and a fall in glucagon. When you do the same experiment, give the same meal to the type 2s, you see a much greater glycemic excursion, a much reduced insulin excursion, and a paradoxical rise in glucagon. So if you go back to the fact that we showed that glucagon is important to glucose uptake postprandially, clearly this suggests that glucagon may be playing a significant part in that abnormal hyperglycemic excursion. One study in which this was more directly assessed, because it's hard to differentiate what occurred because of insulin problems versus glucagon problems in the previous group, this was an attempt by Bob Rietz's group to try and separate the two out. And so these are clamp studies in type 2s, and what they're going to do is, and if you look at the bottom first, they're either going to mimic the abnormal type 2 glucagon profile or the normal glucagon profile. And they're going to try and do it with matched insulin after giving a glucose drink. And so the readout is going to be, what does the glucose do? And so you can see at the bottom, they created this difference of a gain about 30 picograms per mil in glucagon over that first 2-hour period. And if you look at the top, clearly when the glucagon was low, the glycemic excursion was reduced. Now, they didn't hit the insulin match perfectly. If they had actually matched the insulins, if the dark green had been up where the whites were, the glucose level would probably have been even lower. So this is kind of a direct proof that, in fact, glucagon makes a difference under circumstances like this. So, glucagon is abnormal in both type 1 and type 2s, and the data suggests that it is contributing significantly to the metabolic phenotype. I show you data now from a mouse model published again out of Roger Unger's area a couple of years ago where they basically made mice diabetic, either using streptozotocin or aloxan. And they're going to look at this in animals that either have normal glucagon receptors or have had them knocked out. And what you can see is what we all know. If you give these compounds to mice, you will rapidly kill their beta cells, and they will become massively hyperglycemic, ketotic, and ultimately die. If you do exactly the same thing, but you just don't have glucagon receptors around, the mice are fine. Now, these mice have other major abnormalities. Their glucagon levels are out the roof. Their GLP-1 levels are abnormal, FGF21. So I wouldn't read too much into this physiologically, but I think it does underscore the power of glucagon to make a difference in this circumstance. Now, what aspect of knocking out the glucagon receptor has actually caused this improvement, I think, is still open to debate. Let me finish by looking at a few examples of how we might attack glucagon therapeutically in the diabetic. So clearly, if you think about this, there are two major ways you can approach it. One is to control glucagon secretion somehow, so lower it. The other would be to antagonize glucagon action somehow. And both approaches are being actively investigated as we speak. This slide shows data from a study we did in collaboration with Novo Nordisk using a small molecule glucagon receptor antagonist. And I show you it simply to point out that when you give this, and the experiment had a 40-minute control period, and then we did a euglycemic clamp, and what you can see is the insulin level was the same in the two groups, but in the group where we gave the glucagon receptor antagonist, the glucagon levels began to rise. And that is a universal finding. If you inhibit the glucagon receptor, glucagon levels in blood will rise, you will get alpha cell hyperplasia, and somehow there is a signal sent, presumably from the liver to the alpha cell, which changes its biology quite dramatically. But what I also want to show you is that we clamp the glucose here, and what you can see is what happened to net hepatic glucose output. And clearly, the antagonist, I should say, was effective, inhibiting glucose production by about 50%. So it's possible to design small molecules which will go in and turn off glucagon action. This is data from a study done at Merck, where they also had a whole series of small molecules. And these are data in the mouse and the monkey. This is a humanized mouse model and a normal monkey model. And basically what you can see is the gray is the vehicle, the green is where they just give glucagon without the antagonist, and the red is where they include an antagonist as well. So both in primates and rodents, certainly molecules can be built which will do this. Now, I would say that in general, companies that have taken this path have had problems with small problems with lipid metabolism and perhaps some liver enzyme issues, perhaps secondary to inflammation, the nature of which is probably not surprising given what I told you earlier, but and a little unclear as to kind of what that means, but it has slowed up development. Another approach shown here is to play with the peptide itself, so make a change in glucagon to make an antagonist, basically. And these are data from a cell line that was infected with the glucagon receptor. And what you can see is the white bar is just the baseline. The gray is what happens when you give glucagon. The dark greens are what happens when you give the modified glucagon. In the absence of glucagon, doesn't do much. And then what happens if you combine them? So looking at the ability of the modified glucagon to arrest the effect of actual glucagon. And you can see there's a nice dose response. So yet another way to do that. Here's data where they've used an antibody to the glucagon receptor to try and control glucose. And this is an IPGTT in a diet-induced obesity fed mouse that was a little bit obese. And you can see, again, knocking glucagon out markedly improved glycemia. And then lastly, using a genetic approach to modify the glucagon receptor, so using siRNA to look and see what happened. And you can see on the top, you get a pretty good knockdown of the message. As I've mentioned, when you decrease the glucagon receptor, either decrease it or block it, you get this rising glucagon, which is what you see in the upper right. But what you can also see in the two lower panels is both the fasting and the ambient, meaning just taken during the feeding cycle, there's a marked improvement in glucose. So bottom line, there are now a whole array of ways to inhibit glucagon action. Small molecules, modified peptides, antibodies, genetic approaches, they all improve glucose dramatically, and it remains to be seen what the problems are associated with them. They clearly all cause glucagon to go up, probably meaning that there is some hyperplasia. And so I think it remains to be seen whether this approach is going to prove effective or not. Right now, I think we've developed tools to answer the question, and it remains to be seen whether the side effects associated, the adverse events associated with this approach can be overcome. I think in view of time, I will miss that one and go to the next slide. So the other approach is to modify secretion, and there is an awful lot of work being done right now in that regard. I'm going to show you one thing here just to illustrate the point, and these are individuals that were either given placebo or vildagliptin, so a DPP-4 inhibitor. And if you look at the bottom panel, and there's a meal test going on, you can see that the blood glucose rises and then falls. GLP-1 in the presence of the vildagliptin is obviously enhanced. And one of the things that does, in addition to causing insulin secretion, is to limit glucagon secretion. And so you can see in the middle panel the inhibition of glucagon. And most studies that have been done would suggest, I think, that the inhibition of glucagon is probably equally important to the rise in insulin, to the efficacy of these compounds. And I don't have time to go into the data, but if you were to look at some of the incretin mimetics, so Exendin, others, you would see a similar inhibition of glucagon, which I think contributes to their overall efficacy. Maury Birnbaum published a paper in Nature a year or so ago, which suggested actually that the way metformin works is to interfere with glucagon's ability to activate adenylate cyclase. So it's conceivable glucagon may even work in that drug's mechanism of action as well. But there's also, I think, a big push to look at other ways to control it. And certainly that can be done by looking at somatostatin. The somatostatin receptor on the alpha cell is reasonably specific. It's the number 2 receptor, and so they're looking for agonists that would selectively inhibit glucagon secretion. They're doing a lot of work with GPRs, where some of them have been found to be potential regulators of glucagon secretion. So I think this is a rapidly emerging area, and perhaps takes on more significance from the fact that there have been now at least 2 papers that suggest that, in fact, the alpha cell defect precedes the beta cell defect in a type 2 individual. There was a very interesting paper published in Cell Metabolism back in April, suggesting that kisspeptin, which is a peptide that comes from the liver, under the influence of glucagon, can potentially affect and make the beta cell dysfunctional. So the idea would be that the alpha cell works too hard, you get an over-secretion of glucagon, an overactivation of the liver, and that drives then the defect in the beta cell. And there was a paper actually in AJP published by Jameson et al. A number of years ago, which also suggested that the alpha cell goes wrong first in type 2 diabetics. So, in summary, what I hope I've been able to show you is that glucagon sensitively and rapidly stimulates hepatic glucose production. And that's key for its physiologic role. Its acute effects depend primarily on glycogenolysis. I'm sure chronically jazzing up the liver's gluconeogenic machinery in a circumstance where you can, for some other reason, supply precursors could be important, but that remains to be defined. Glucagon impacts glucose uptake by the liver and thereby can contribute to postprandial hyperglycemia and glycogen storage. It is not simply a hormone of glucose release by the liver. It is very intimately involved in glucose storage. Glucagon is the first line of defense against hypo, and this is lost in individuals with type 1 diabetes. Anything that we can do to try and understand that loss and correct it would be therapeutically very beneficial. And lastly, relative or absolute glucagon excess contributes significantly to the metabolic phenotype of both type 1 and type 2 diabetes. And I think we're making rapid and dramatic progress in understanding ways to be able to manipulate glucagon. And if we can do that, I'm sure we can dramatically impact therapy. The last slide just pays homage to all of the people who did our piece of the work. Thank you. I'd be happy to spend 10 minutes or so answering questions should anybody have them. Thanks a lot.
Great. Thank you so much for a very interesting presentation. We actually do have some questions from our audience, so I'll start with the first one, and it is, could you please comment on the role of bile acids in relation to glucagon?
Well, it's a very good question. I mean, the whole role of FGF21 and FXR in glucagon physiology and by definition the bile acids, I think is really unclear. I think this is another area where knowledge is expanding incredibly fast, and it's not something that I'm comfortable at this point drawing any clear conclusions on, I'm afraid.
Okay, the second question is, in your pancreatic clamp experiments, it seems that upon glucagon infusion, hepatic glucose production goes up quickly, resulting in increases in arterial glucose. However, hepatic glucose production falls quickly during glucagon administration, yet arterial glucose levels remain high. Metformin has recently been postulated to antagonize glucagon in the liver. According to this hypothesis, how does glucagon antagonism mediate metformin's pharmacodynamics, that is, reduce insulin resistance and normalize blood glucose?
Again, I'm not sure. There are so many ways that metformin has been postulated to affect the liver, only one of which is through metformin. And there, of course, the idea is that the AMP level is driven up by the glucagon, and when the AMP level is up, that inhibits the amylase cyclase, and so you just decrease overall glucose production. I'm not sure quite what you mean by the time course, but let me make a comment on that anyway, because I think one of the intriguing things about glucagon is that basal levels have a constant effect. I showed you that withdrawing glucagon has an immediate impact, and it actually doesn't matter when you do that, whether you do it after an overnight fast, after a prolonged fast. If you give and elevate glucagon and let the body downregulate and redefine it, and then you withdraw it, it also has a continual effect. So it's got this crazy stable effect at baseline levels, but it is downregulated whenever it's elevated. Now, teleologically, that's probably to prevent an excessive hyperglycemia. As to what the mechanism is, obviously, if insulin and glucose can rise, they will offset glucagon's action. But if you control for them, as we did, then it still comes down. Now, you can control for the hyperglycemia, but even then, you'll see about a 50% tachyphylaxis over time. And there are several thoughts as to what that is. The first is an activation of phosphodiesterase in the liver, which causes cyclic AMP levels to decline, and certainly they do decline, and they decline rapidly. But in our hands, they don't decline enough to reach a rate-limiting point, so we actually don't think it's that. There are two, I think, current circulating hypotheses to explain it. One is the fact that the G6P levels go up, and that, in fact, is what is driving the inhibition of glucose production. The second is out of Tony Lamb's lab, where he has basically suggested that the brain is glucagon sensitive, and that what happens is you, through the parasympathetic nervous system, drive an inhibitory signal to the liver, and that turns down glucose production. So its central effect limits its direct effect on the liver. And he further suggests that that may go wrong in individuals that are obese or overweight.
Okay, and moving on to our next question. One application of glucagon is dual hormone artificial pancreas, where glucagon is infused in type 1 diabetes in a continuous manner along with insulin. Do you see any negative effects of using glucagon in this way?
So again, I think that this is an emerging area. Clearly, there have been a number of studies published recently showing that if you basically have a joint infusion of insulin and glucagon, you get better glycemic control. And the theory for that is, with glucagon being elevated, your chance of hypo is lessened, so you can be a little more aggressive with your insulin infusion, which also then lessens hyperglycemia. So the oscillations, the range of oscillations actually changes. I don't really see a problem with that. I think, you would want to keep your eye on triglycerides and lipids just because glucagon has an effect on those. The other thing that glucagon does very sensitively is regulate the level of amino acids in blood. So I think keeping your eye on plasma lipids and plasma amino acids is probably useful in experiments that are done to test this out. But I don't see any real reason why why there would be any unanticipated problems, because the glucagon is only, I don't think it's, at least my understanding of it, is not chronically infused. It's infused only in response to a fall in the glucose level to a certain point. And so I think that's different than if it were to be chronically infused, where there could be other issues.
Okay, and it looks like this next one is our last question, and it is, given glucagon can act on both the glucagon receptor and GLP-1 receptor, would you please comment on the glucagon effect on GLP-1 release and action?
Well, that's a very good question, and I'm not really sure that I know the answer, to be honest with you. Certainly GLP-1 can inhibit glucagon release. The problem we have in getting data that assess glucagon's effect on GLP-1 is that when we use the pancreatic clamp, the somatostatin inhibits the release of GLP-1. So we really don't have any data that would allow me to answer that question, and I'm not sure what data are out there. It's not an area that I'm familiar with.
Well, I'd like to thank all of you for attending today and submitting your questions, and a very special thank you to Dr. Allen.
Thank you.